Welcome to the Earthworks Podcast, where our team will share the jargon of carbon from many of our turf friends from the past 30 years.
Hi, everybody. It's Joel Simmons. I'm sitting here with Jack Higgins, and we have Mr. Science with us, Lawrence Mayhew, who is our chemist here at Earthworks. We've talked with a number of times here on the Earthworks Podcast, and yes, today we have him back. And we want to talk about today the myth of the 16 essential elements. And we've been talking a lot about this on our Monday morning meetings with Lawrence. But Lawrence, I'm going to kind of let you run with this. And let me just go a little bit further introduction of Lawrence. Lawrence has been working with Jerry and or me for a very long time. I'm not even sure I know your full story, but I do know that you are a world-renowned expert on humic substances, and we've been relying on you for that for a very, very long time. And in full disclosure, Lawrence works very closely with us on formulation of products, helping us get the best products, certainly in the liquid side that we can possibly have. But more importantly, Lawrence, we lean on you. But this conversation started a while back when we were meeting on Monday morning. And it's come up a number of times, at least internally, that this old theory that the plant, the only essential elements are the 16 essentials. Do me a favor and let's start the conversation with how the hell did that even get started? I mean, I think you told me the story. It was done in the 30s in a hydroponic environment. But I mean, you know, I think, and you can correct me if I'm wrong, but I think most, progressive scientists, if I can use that phrase, realize that that's kind of bunk. I mean, obviously the plant's using a lot more than 16 elements, but start with where that whole concept got started. And then let's kind of build out the conversation on what the plant really is looking at internally.
Arnon and Stout, 1939. They published an article on on nutrient solution. That'd be a solution you put together in the laboratory in a beaker, set a plant in it, and then the plant's supposed to grow. And what they did was they started eliminating elements. And then when they found out that if the element nitrogen, for example, was removed from the soil, I keep saying soil solution, from the nutrient solution. I don't work with nutrient solutions. If they remove nitrogen, for example, from the nutrient solution, or if they remove potassium, or if they remove phosphorus and left everything else in that solution and the plant died, then they said, oh, that's essential. So a plant has to have that. They came up with less than 16 back in 1939 because they didn't know about micronutrients as we know now. So they came up with a very short list of nutrients that are absolutely required by plants or they die. So that's like saying, let's put a bunch of sailors on a ship and let's try to discover whether the world is round. Let's say the year 1517, and we'll hire this guy called Magellan to run this boat around there. They get halfway around the world and the crew is dying from a vitamin C deficiency, but it's not essential. You can live without vitamin C, but the crew, 17 crew members made it back out of 3 ships.
A little scurvy, eh?
Yeah, dying from scurvy. But hey, vitamin C is not essential. You can take vitamin C away from a plant. You can take it away from an animal. They will survive. They just don't die. So that's the definition of essential elements in plants, and that's the history.
So, but you said there were less than 16 elements. I thought the number was always, 16. I still hear this in conversation in our industry that, there's only 16. I mean, I remember one time on a Twitter feed we were talking about how many elements are in some of our liquids, and there's as many as 75. And people would come back, oh, that's a bunch of bunk, and there's only 16 that matter, and blah blah blah. And that's where this whole conversation started. Is
okay.
That obviously.
Well, it's worse than that.
Great. Before we get going, let me let everybody know that you are showing us a whole bunch of slides. So if there's a chance you can see this on our YouTube station, you can actually see some of the work that Lawrence is highlighting. And we'll talk about it as best we can on the audio side. But if you do have an opportunity to jump over to the YouTube side, you can actually see a PowerPoint presentation that Lawrence is going to go through here this morning. So
Okay.
I'll let you have it back, Lawrence.
So I'm on the screen right now is a typical soil analysis report from a very famous lab in the Midwest. And they only list 11 elements on that laboratory report. So that's what I meant by it's worse than 16.
It's
When you get a lab
11.
report from a typical laboratory, you only get a report on 11 elements.
So you got the
And
basics. You got phosphorus, potassium, magnesium, calcium, sodium, and then we have sulfur, zinc, manganese, iron, copper, and boron. Did I miss anything? Well, there's a percentage on hydrogen, so maybe you can
Right.
add that one in there. But
Another
that's
thing that these labs typically do when they give you a report is if you could see this, it'll say, oh, the phosphorus is medium, the magnesium is very high, the potassium is low, and that there's low, high, medium, all the way across here on what they think are low, high, medium. Guess what? That's absolutely arbitrary. There's no scientific basis to say that that is high, that is low. All of these reports are based upon a history. You're always asking, how did things get started? This got started with extension agents telling these labs, these commercial labs, what they should test for and what the numbers should be. So they dictated that back in the 1930s and 40s, and that has become pretty well standard in our industry. So there is no scientific peer review studies on whether that's a very high magnesium or that's a very low potassium, et cetera, et cetera. Interesting. So the background doesn't even exist to support these reports. These
So you're saying this is subjective, that these numbers are
subjective, and they vary from lab to lab. You can send the same sample to a different lab and get a different report, different recommendation. So it's whatever you're used to working with that it comes down to.
And this is why we always say that a soil test is really a roadmap and not a report card, because like you said, I mean, we can take the same sample in any laboratory. Obviously, we're working with Logan Labs, but any laboratory, if you tested the same sample, you'd get a different. You would extract a little bit different each time. But when you look at this particular test, you've got a CEC here of almost 12. So, you would think that those numbers, high and low, would still be based off of the holding capacity of that soil. So it's kind of...
they're based off of whatever the university extension agents told that lab they should say. And you can call them up and talk to Jim Fashing at Midwest and ask him that question. That is the answer. Jim Fashing told me this personally. Maybe he won't share that, but he's on the verge of retiring, so that's probably...
Sorry, Jim. All right.
Now, another point is that none of these elemental reports actually correlate quantitatively to how much a plant will take up. There is absolutely no correlation between what it says on this report and what the plant will take up, because the absolute numbers don't matter. Only the ratios of these numbers really matter, which is called balance. I mean, 11 elements? Seriously?
Yeah.
Okay. The state fertilizer laws say that there's 16 essential elements, and here's the list. And N, P, and K. Now look at the P2O5, 1%. 1% guarantee of phosphate, I can guarantee you, is an ecological disaster. To put that much phosphate into a soluble form and apply it at a 1% rate is like orders of magnitude more than how much phosphate there is actually in a soil.
What you're saying is, if we were to apply that much phosphorus, is that what you're suggesting?
I'm saying that if you have a fertilizer and it's guaranteeing that 1% of that fertilizer is P2O5, that is environmentally unsound practice right there. That's what I'm saying.
What would a normal fertilizer be in terms of percentage of phosphorus?
Oh, you can jack that number up into the 20s and 30s if you want to get a high phosphate fertilizer, which just, to me, these are indications as to their environmental impact. So if you had a 111 as it shows on here, that'd be a very low environmental impact product compared to numbers that are much, much higher than that. And I rarely deal in highly soluble phosphate or potash or nitrogen.
Yeah, so a product like MAP is what, 1152O?
Yeah. Yeah.
So
It's incredible. Absolutely incredible.
Let me read through this list just so everybody that's listening sees it. So we're dealing the 16 essential elements as listed by the 1934 study: nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, boron, chlorine, cobalt, copper, iron, manganese, molybdenum, nickel, sodium, and zinc, all at varying rates. I mean, for example, molybdenum is 0.0005%.
That's a lot of molybdenum
That's
for
a lot.
soil to take.
Yeah.
That's a lot.
Yeah.
Because percentage means parts per 100. Soils do not have concentrations in parts per 100. Soils have typical concentrations of parts per million. That's what your report even says. Or parts per billion or parts per trillion are typical soil concentrations.
So what we're suggesting here is that these are the only elements that the plant, according to this theory, is the only ones that the plant needs. Everything else, and there's 90 minerals on a periodical chart, and 90 minerals that can be shown in a tissue.
You're getting ahead of me.
Yes, I am, but that's where we're going.
That's where we're going.
Right,
okay. Here's the periodic table. This is the standard periodic table as we know it. I highlighted those 16 elements on the periodic table, and visually, if you can see this, they're in the minority. There's a lot more elements out there. Yeah. And so only 16 doesn't really populate this chart very much. So what about the other 102 elements that are on this chart? So let's assume
we don't need them.
No, let's assume for the moment that there is no use for radioactive or synthetic elements, because synthetic elements do not exist in nature. Plants and microbes will not recognize them as anything. So let's take all those away, and we end up with a periodic table that's shrinking. Okay, so I got rid of all the radioactive and synthetic elements. Okay, what about inert elements? They're gone. They're gone off this chart. That'd be like neon, argon, krypton, xenon, radon. Those are elements that do not interact with anything in nature.
In fact, they're noble.
And let's take mercury out of here, because I'm not aware of any evidence that mercury is needed for plants or microbes. There seems to be no use for mercury, so I took that off the chart. As a result, we end up with 75 elements on this periodic table that meet the criteria that I just said. Okay, all 75 elements are found in the rocks of the earth, which produce our soil. Soils come from rocks. They come from minerals. They're found in all soils. They're found in all rocks. They're found in all plant roots to various levels. All of these can be found in plant leaves, and all of these elements can be found in soil microbes. Period. It's just a fact.
Found in soil microbes?
Yeah, you can take a microbe and analyze it just for its elemental content. Yeah. And you will eventually discover that if you analyze enough microbes, all 75 elements will show up in that microbial population.
So if you analyze the human body, I bet you'd find them all as well.
Yeah, let's grind up a human body and look for these elements. You will find all 75 elements.
Yes, let's do that.
Right. The reason why is because the microbes and the plants actually consume these elements from the soils, and then we consume these elements ourselves.
And now that we've been learning about rhizophagy and the work that Dr. White did at Rutgers, these microbes are being brought into the plant so that the plant can harvest all those minerals that the microbe, carries with it, right?
Yep, they're all there.
Isn't that essentially what he's suggesting in that research of rhizophagy?
What I get out of his research is that there's this amazing thing going on all this time that we didn't even know about. Mm-hmm. That's what really gets me. It never dawned on me in all my years of research that plant roots are actually eating microbes.
Yeah.
And then spitting them back out again and saying, come back. We're going to do this again and again and again and again.
All right. So you've identified 75 elements here that
yes,
potentially are being used by the plant. Is that the theory?
Not potentially.
Well,
I'm just saying right now, I'm just saying, look, if you look at all the rocks of the earth and all the soils derived from the rocks, and you grind up plant roots and you analyze them, you grind up plant leaves, you grind up microbes, you grind up humans, you will find all 75 elements. Now, there's some might be trace or ultra trace quantities, some might be in major quantities. I mean, the quantities really don't matter. It's the balance of these things that are found in nature. In nature, there's this thing called ecological balance, and the elements are definitely part of that balancing system.
You're going to get us in trouble with that word balance. Some of the boys hate that.
Well, you know what? Then let's use the right word. Ecological stoichiometry.
Which is a description used to actually indicate what the true balance of materials are out there. In other words, you can analyze all the scandium in all the soils of the face of the earth, and you can say, well, how much scandium is there relative to calcium, for example? And that's a legitimate thing. That's done by scientists all the time, by geologists. Geologists understand these balances. They know what's normal out there. They know when these balances are off. There's a lot of ways of looking at balance. Anyway, I better keep moving.
Yeah.
Okay. The science of looking at each and every one of these elements and asking the question, what do they do in biological systems, is actually the science of elementomics. Okay, so there is a science. Yeah, there is a science that looks at these, every one of these, and it tries to discover what is their active role in biological systems. That's the question. That's the basis of elementomics. And I drew a line from the extreme right-hand side of this chart, everything to the left of this periodic table, which is overwhelming majority, 64 of the elements on this table are metals. Okay, just kind of set that aside for a minute. Metals are very reactive materials chemically, so they all have the ability to act like metals. And what we mean by metals is their ability to actually share electrons. Okay. Scientists can use what's called geochemical analysis, and that geochemical analysis is used to detect all 75 of the elements I'm talking about. So in other words, a typical soil report is unable to extract out these elements. But if you take a soil sample or a leaf sample or anything, you treat it with geochemical analysis, which are very harsh reagents to extract out these elements, you will detect all 75 elements. Now, going forward, I want you to keep this in mind, because I read this 25 years ago and I was blown away by it. This is a quote by Professor Silver, University of Chicago, and it was published in a book that I have here that was published in Madison, Wisconsin. The book is called The Geomicrobiology Interactions Between Microbes and Minerals. It's a review by the Mineralogical Society of America, and the quote is, "Nothing in nature is uncontrolled. Nothing is left to chance."
Wow.
Love it.
That became the basis of my research. Pardon?
What year was that printed?
1997.
So certainly in our lifetime. So, I mean, this has been a conversation that's been going on for a very, very long time.
Very long time. And when this book was published by the Mineralogical Society of America in Washington, D.C., it's number 35, issue number 35. I lost my copy, or I destroyed it or something. So I called them and I said, I need another one. They had a backlog of this issue. One of the most popular issues ever published by the Mineralogical Society of America.
So let's read that again. Nothing is left.
Nothing in nature is uncontrolled.
Nothing
is left to chance. I mean, I didn't say this. He said it, and it just changed my career. I just thought, oh my gosh, that means we've been told all these years that a plant will take up something totally by accident. It just can't help itself. It's beyond its control. Right. Yeah. Well, this guy totally disagrees with that.
And that's really the crux of this conversation, is that nothing is uncontrolled here. I mean, the plants will take up the minerals because they, for a purpose. I mean, maybe we don't know the purpose, but it's not left to chance.
That's true. So going forward with that, I discovered this series of books down in the archives at the University of Wisconsin, and I went out and bought my own. The book right on top is, okay, this is a series of books and it was published by Marcel Decker, very famous, very reputable publishing company. The series of this books was called Metal Ions in Biological Systems. Now, remember, I just said 64 of those elements on the periodic table that are found in plants, minerals, and microbes are all metals. And this whole series was on metals. The book right on top says vanadium and its role in life. Did you ever see vanadium guaranteed on a label?
Not even sure I know what it is, to be honest with you.
Okay, the second volume is about interactions, how these metals interact. Third volume is their biochemistry and availability and transport of these metals. That's a lot different than looking strictly at soluble materials. Chapter number 40, book number 40, issue 40 is the lanthanides and their interrelations with the biosystems. The lanthanide group on my periodic table I showed you earlier were 14 elements sitting down at the bottom of the table. Then the last book on this photograph is molybdenum. Okay, we know a little bit about that, necessary for nitrogen utilization, and tungsten and their roles in biological process.
And that's the biggest book of this collection. Is that really true? Is that just the
yeah.
big...
Yeah. Tungsten, my gosh.
That book is huge. I'm sure that that's an easy read, all right?
Oh, yeah. I took a deep dive into these. I mean, I never heard of tungsten being in biological systems. Turns out, pretty critical material.
You know, Jerry Brunetti was very big on molybdenum, and he spoke
molybdenum
about
and cobalt.
And cobalt, and he spoke a lot about it, and I'm sure the two of you had gone back and forth on this a few times, but molybdenum has been something that we've always focused on because of the work that Jerry did, and cobalt. We certainly promote it in products. So kind of interesting to see, going full circle here, seeing some of the research on this.
Well, absolutely necessary for nitrogen fixation.
Yeah. Molybdenum.
Okay, I'm showing a result of an analysis that I ran, I don't know when it was, 25 years ago, on a natural salt. And this salt came out of a mine in Kansas. And I just ran an analysis on the salt. And this is kind of where I got my start looking at trace and ultratrace elements.
Wow.
And this really got my attention. I just highlighted all 14 lanthanides up on the screen here. Cerium, dysprosium, europium, gadolinium. Gadolinium is found in MNR units and in all kinds of medical equipment. Lanthanum, lutetium, neodymium, rare earth magnets, praseodymium, samarium, yttrium, and ytterbium. Now, all 14 of these, it turns out, are extremely interesting. And the research is now becoming more and more available about what they do in biological systems. Oh, yeah. The reason why I put this up there, somebody said there's only 16 that count. I got 14 on the screen right now. I don't know if you're keeping count or not.
And they're not even on the list. Explain again the group. What's the name of this grouping?
The lanthanides.
And explain
more
that.
More popularly known as the rare earths.
Okay. So these are the rare earth minerals. That are not even part of the traditional 16 elements that the plant needs for.
Totally overlooked.
Yeah. Okay.
These elements are very important to regulating calcium, especially when there is stress. Name a stress, these elements will substitute for calcium in any biological system, including humans. They will interchange for calcium. We really don't know why it would interchange samarium instead of cerium. The science is just starting on this, but the interchangeability of these is very easy to study. And their chemistry is so similar to calcium that they interchange with calcium. Very important to calcium utilization.
So what you're suggesting is if there's a soil that is deficient of calcium, the plant is under a level of stress where it knows it needs to bring calcium in. It can't find it or bioactivate it enough to get it in. Any of these metals, these rare earth metals, could substitute for the calcium intracellularly. Am I?
That seems like a jump to me.
That would be under what we would call sufficient or insufficient conditions. In sufficient conditions, in other words, you have sufficient calcium, you have sufficient lanthanides in the soil, they'll interchange anyway if there
Right.
is stress present.
Right. So, and we don't really know why, or is there a hypothesis?
Oh, yeah. Well, it's because they are much more, they have a higher chemical activity than calcium.
Mhm.
So if calcium is deficient in the plant, and the plant wants to really ramp up the benefits of calcium, it will seek out and obtain any number of these lanthanides.
So yeah, the calcium or the plant uses calcium for a number of different benefits.
Yes.
We'll rattle off some like just strengthen cell walls, defend structure,
Mhm.
that sort of thing. Build cells, like grow roots, that sort of thing. We can consider these rare earth metals like a super calcium in terms of delivering those benefits.
That's an interesting term.
They're like it's like calcium with a
He
cape
just
on
made
it.
it up, but I like it.
Yeah.
Yeah.
So
Yeah.
does that make sense?
Yes.
Okay, so these are like supercharged calcium. And are those the sort of benefits that you're talking about? That structural benefit?
Structural all the way to stress, even frost resistance. Calcium is renowned for avoiding all the issues with frost, for example, which is a cold stress placed on the plant. And when that happens, the cells will actually they will exchange their calcium for any of these lanthanides, because calcium at those extremely low temperatures is not as bioactive as these materials are, and that's why they interchange. This was discovered in clams. That was the first discovery about these things interchanging. There was ocean studies that discovered this
Oh,
some
discovered
time
in
back.
clams.
Yeah, clams, because they have calmodulin in them, which is the signaling mechanism of calcium. Calmodulin. I don't know if you guys studied that or not.
Never heard of it.
Nope.
Calmodulin.
That's why we have you.
So
Okay,
that-
calmodulin is a secondary messenger that runs throughout all biological systems. That means I'm including humans, all the way down to microbes, plants, everything. Calmodulin is the signaling mechanism that runs through biological systems to say, wait a minute, we're too cold, we're too hot, we're too dry, we're too wet. And that signaling mechanism is a means of communication. And they found out that they end up calling it landmodulin after lanthanum. They found out that substitution was taking place in clams, and they discovered it was happening in almost all biological systems.
Right. So the signaler could say the calmodulin signal metabolite that you're saying could be calling for calcium, or it's saying, wow, the need is so extreme, we need the supercharged. And so let's grab some of these lanthanides, these rare earth minerals.
Yeah, and the calmodulin is signaling to the whole plant that it needs to respond to some kind of stress. And there's a lot of secondary metabolites that get involved in that reaction.
So this goes back to to the concept that the plant understands its needs and has the intelligence to find what it needs when it needs it most.
Yes.
Now,
Where
let
do
me
we
ask
get
you
our
this.
intelligence? Where do we get our intelligence? From genetics, right?
Right.
So where do plants get their intelligence? Genetics.
Shocking.
Genetically driven.
So where do these so in nature, in true nature environments, these minerals are just simply found within the soil structure itself, within the rock material of a soil?
Yeah. Some rocks are enriched in these materials. Some rocks are depleted in these materials. But overall, if you looked at the whole soil system of the earth, you can actually say, well, here's the average concentration of cerium, yttrium, and all this. Yeah, it's most certainly done. I maintain a database of all the elements found in all the soils globally.
And it goes back to that conversation of I mean, you go into the Midwest United States where these soils are big, rich, full of mineral. We can assume that the level of these types of metals are pretty prevalent. I assume that's fair to say.
It really varies from region to region. Yeah, there is a tremendous variation in these. You take any material that's found like in a 1 part per million or lower, or just a few parts per million, then the variation really increases
Now,
compared
Lauren,
to, let's say, calcium.
let me ask you this, because I think a lot of folks will have this on their mind. I mean, we're talking about the need for all these minerals. We've done analysis on our Foliars Plus line, and there's 75, and most of, I think all of these actually are on that list. So if we were to try to supply these types of minerals, where would we get them? Where would a golf course superintendent or a turf manager in particular find, the level of this kind of mineral base in a fertility program?
Well, in our case, in Earthworks case, we need to seek out at least 3 different ingredients that have various levels of these lanthanides in them. And that's how we solve the problem. That's how we bring them to the plant and the microbes, through numerous ingredients. The rock phosphates in northern Canada, Ontario, Canada, are very enriched in these lanthanides. So as a mineral, they can be found in various minerals. But you have to process that. In our case, we have a liquid product that we have to seek out, let's say, ocean water, for example, or something like that, or maybe fish, So we put them all together to get these things into the products.
It goes back to diversity, and this is going back to, when Jerry started this whole formulation process, that we were constantly trying to find as diverse a mineral base. I mean, one of the things that Jerry always talked about is remineralization and finding these minerals. And it's funny you bring up black rock phosphate or rock phosphate, because we've been criticized for it for so long by so many that say, that won't ever become available. But there's so many things in there that the plant, as it spits out its organic acids, can actually dissolve and then find a way to bring it in to the plant material. And I think, you've taught us that that's, being pretty well documented at this point.
Well, that was one of the first things I was told to do when I entered this profession, is go out and find out everything you can about rock phosphates. I probably came pretty close. They are enriched in these materials, and these materials in a phosphate, like you mentioned earlier, MAT, for example, they are depleted in these minerals, even though in the original rock they were there. But they're considered contaminants in a high phosphate fertilizer.
Wow.
And you know how phosphates and calcium, or at least a highly available form of calcium, don't get along too well? Well, they have the same chemistry as calcium, so they are eliminated from any highly processed phosphate fertilizer. They're gone. They're not there anymore.
Oh, and it's because they are so reactive with catalysts.
They're more reactive than calcium, and if calcium and phosphate are reactive, these are even worse. So, they're eliminated. They're processed out.
And nature took care of that issue by putting them in the rock together and kind of like buffering them in mineral form. And then and humans come through and say, no, we have to derive. We have to
reduction.
We only want the pure stuff. I only want pure stuff.
Only need to 14. We're
I learned my lesson on pure stuff by stop eating white rice when I was a teenager. And let's go for the brown rice. There's more goodies in it. Same principle. Yeah. Okay, back to this chart of 75 elements that I found in a salt, just a natural salt. Chromium was highly enriched in this salt, and chromium, it turns out, increases the bioavailability of copper, boron, nickel, and regulates the amount of iron that a plant takes up, and manganese regulates those two very much the same way, and it regulates how much vanadium a plant should be taking up. So in other words, if you don't have any chromium there, these others will not reap the benefit of the presence of chromium. The boron, the nickel, the iron, manganese, and vanadium are not quite, they're actually out of balance at that point, and they're not taken up as efficient as a plant. By the way, plants are more efficient at taking up nutrients when there is an insufficient amount of nutrients versus when there's sufficient or over application of nutrients, the efficiency of elemental uptake in plants decreases dramatically.
No, that's
And they actually get rid of the excess, for example.
So when we are over applying, which is not uncommon in agriculture and any agricultural environment, we actually are blocking the uptake.
You're actually reducing the ability. There's been some really fantastic studies done on nitrogen and potassium in that regard. One of the studies was called the potassium, what do they call it? Paradox. The more potassium you put on, this is scientific fact, by the way, the more you apply potassium, especially potassium chloride, the less potassium is taken up by the plant, because the plant begins to expend its energy trying to spit out all that damn potassium that's getting into it. It doesn't need that much, and that burns up a lot of energy. So then the plant ends up kind of overreacting, and it takes up less potassium than when potassium was insufficient. Interesting studies, fantastic studies. Same thing for nitrogen. These studies were done back in the 80s and 90s. They were very well.
Certainly in our industry, nitrogen tends to be over applied. I mean, a lot of things are over applied, and we've been talking for years about, helping to build a biological environment.
Way over applied.
Exactly.
If they didn't, we wouldn't see our oceans ruined like we are.
Right. The bay. Yeah.
Okay. Another point. This study that I was doing on this salt, I noticed for the first time in my career that the strontium was heavily enriched relative to calcium. So I dug into that. I thought that's not by chance, right? Silver, according to silver, nothing occurs by chance. Then I found out that in every naturally occurring mineral except limestone, calcium and strontium are always very, very well coordinated. Why is that? Strontium interchanges with calcium again when there's stress or when when there's insufficient amounts of calcium, strontium will take over. So whoever heard of strontium being in a fertilizer? But these are the kind of things I look for when we put together a fertilizer.
Wow. So obviously not one of the 16 essential elements.
Definitely not on the list of 16. Now, I also see the reason why I'm putting this results of this salt up here, because it kicked off a whole new way of looking at balance. And then from this point on, I noticed that whenever potassium in a naturally occurring soil or a naturally occurring mineral is enriched, so is rubidium. And guess what? Rubidium and potassium interchange with each other also. So there's this pairing throughout nature. If you have this, you always have that. If you have this, you always have that. Tit for tat, all the way down the line. These are the majors. Calcium, potassium, they have their counterparts. Calcium has its strontium, potassium has its rubidium. You take away either one of these out of a system, and they will substitute for each other. You put them together in a balanced, like this ratio right here, for example. Just look at that ratio. Phosphorus requires nickel, and it also requires strontium in order to become an efficient uptake mechanism. So the mechanisms that plants use are energy driven, they're genetically driven, and they need more than just one thing in order to pull it off. They need pairs, they need complements, they need complementary minerals. Cobalt is very interesting, and molybdenum, as you said, Jerry Bernetti studied these very, very deeply. But nickel, I never heard Jerry talk about nickel, but nickel is out there. It's now considered essential. Strontium, titanium, tungsten, vanadium. What all these have in common? The absolute requirement by nitrogen-fixing microbes of all these that I just listed. So nickel, titanium, tungsten, and vanadium, strontium, molybdenum.
When you say nickel is now essential, according to whom?
APCO, Association of American Plant Food Control Officials.
I did not know that AFCO had that in there. That's good. So AFCO
came out of the pecan industry. The pecan industry said if we don't put nickel down, we have huge disease problems and developmental problems in our trees and our nuts.
Wow.
And they approached AFCO. I was there when the industry approached AFCO and said, we want this to go down as one of the elements that we can put on a label. They had to bring in USDA scientists to prove their point. They did.
Just so everybody understands AFCO, explain AFCO. Who is AFCO?
AFCO, all of the fertilizer laws in the United States are state laws. There are no federal laws, with the exception of the organic rules, but all fertilizer laws are regulated state by state. And the state regulators gather together every 6 months at some place of their choosing, and they have a meeting and they talk about things like this. And APCO stands for the American Association of Plant Food Control Officials. And I always say there's no such thing as plant food, but anyway.
And since you've been involved in AFCO, how many minerals have they brought to the table as now being accepted that you can call? I mean, certainly more than the original 16.
16. They're still there. They're still stuck at 16.
But nickel's now approved, and you're saying that
1939 it was less than 16.
But silicon, you're saying, you were telling us the other day, silicon is about to be approved. Nickel's been approved. There's been others that have been approved.
AFCO is slowly beginning to realize that they're just starting to begin to to perhaps understand that there are other elements besides the 16 that they list. And they're now moving them into a category called beneficial elements instead of essential.
And what's your silver ball saying in another 10, 20 years? Will we see more hit this list? I mean, you did mention a silicon probably will hit that list soon.
It depends on the size of the industry and how deep their pocketbooks are, because if you want to start something rolling through APCO, you're looking at 6 years and endless meetings. And it's very, very, very expensive. It took 6 years for us, for the Humic Products Trade Association, to get humic acids and hydrophobic acids approved by APCO. It took 6 years. And I complained that was way too long, and everybody laughed at me and said that I was on the fast track. So
6 years was the fast track.
Yeah.
Wow, that sucks.
Okay. So point being, I don't know if anybody's counting, but we're definitely over 16 by now. Okay, enzymes are proteins. Proteins are critical to the function of everything in plants and microbes and in people. But enzymes are a class of proteins that act like catalysts, and they are in our biological systems to speed up a specific reaction. There's a specific enzyme for a specific reaction. I got a picture of a racehorse and a jockey on the bottom of this slide. The point is, when you want that horse to go, you giddy up and go, right? You slap him on the butt. You do something. In other words, that whole biological system strapped between the legs of that jockey has just totally changed everything, and it needs something, and it needs it now, and needs it fast. Now, what happens at the checkered flag? I don't know if they have checkered flags in horse racing, but they sure do in auto racing. So at the checkered flag, what do you do then? You got to back off, right? You just can't go full bore relying on these catalysts to speed up every reaction when you don't need it anymore. Okay, the point is, in order to pull that off, enzyme cofactors are required to speed up or to slow down these enzymatic reactions. They just don't go full blast 100% of the time. Nothing can survive that way. These cofactors are almost always trace elements or ultra trace elements in these biological systems. So that's going to require a definition. I'm going to call them trace and ultra trace elements. The trace and ultra trace elements are also used to slow down these reactions. That was my point. You want them to get in there and speed things up, but you need to have a totally different element back in that enzyme to slow things down. Okay, so these trace and ultra trace elements are renowned for actually modulating the activity of enzymes. I didn't make that up. Look at this. This is one of my favorite references, Encyclopedia of Metalloproteins by Springer. Very, very famous publisher. My photograph here, for those of you who can't see it, is a stack of reference books that are stacked 4 high, and it's encyclopedia. They're extremely thick. You just pick any of those elements that I've been talking about and look them up in the Encyclopedia of Metalloproteins, and you will find out what their function is in a protein or in an enzyme. There it is. There's the reference right there.
And you've got a whole bunch of tags in that book that you must.
Yeah. Probably vanadium.
This might be a good time to talk about this because, when we built the product, and I'm going to be self-serving here and I apologize, but when we built the product Protein Plus, Jerry and I put this together with the idea, and the reason why the product was called Protein Plus was because it brought to all the table these cofactors that you're talking about. Jerry understood that. We put together a product that had all the cofactors so that when all of these materials were brought into the plant, it could quickly build an amino acid, protein. And can you help us better understand that process and what Jerry, was able to accomplish when we put this together?
All proteins have nitrogen in them as part of a very complex and extremely folds on folds on folds of molecular structure. And if you were to pull all those folds back, you not only find nitrogen, you also find these trace and ultra trace elements, or you might find a major element in there. For example, photosynthesis is dependent upon magnesium, so you will find magnesium folded into that protein for photosynthesis. The point is, if you do not address every aspect of the elemental requirement of a plant, in other words, somehow you do not supply it with these trace and ultra trace elements, the proteins cannot be processed by these systems to the point where they become proteins. They will languish. They will remain either as amino acids. They won't form peptide bonds. They will end up being something in between an amino acid or a complete protein. The complete protein is what the plant needs. And if you do not produce complete proteins, you invite insect attack, for example. Complete proteins are not required by insects, but insects do like amino acids or incomplete proteins. So this whole process eventually ends up making proteins. That's kind of the end product. That's what we're
And the
really
key
all
thing
about.
here is that we're providing all these cofactors, whereas if traditional, agriculture or even turf management would say, let's just put down a urea, a straight nitrogen.
Sure, that's all we need is urea.
Right. We've heard that so many times and recently, unfortunately. But, in the process, the plant brings up the nitrogen and it sits waiting
that just
for everything else.
barely keeps me from cussing. But anyway, I won't. I'm being recorded.
We're allowed to cuss on podcasts, I think. But
So
Oh, really?
we do talk about, yes, we provide those cofactor, trace, and ultra trace elements. Do they come from the whole array of amino acids that are in that product and in all of the biocharge liquid products?
Could you rephrase that?
So we're talking about Protein Plus, and we're talking about providing all the cofactors for building a complete protein. But I want to tie that into the amino acids that we have in the products.
Oh yeah, I jumped. Okay. I got to go back to high school science class.
Please, let's do that.
Okay, back in high school, we were told that amino acids are the building blocks of proteins, and that's true. The amino acids combine with each other through what's called peptide bonds. I think I just threw that out there a few minutes ago, like everybody knows what that is. So these peptide bonds rely upon enzymatic activity in order to go forward. So once you build amino acids upon amino acids to produce proteins, then that's the way you get proteins. Amino acids are the building blocks.
So if they're in the formula, do they already have a lot of those cofactors that we're talking about within the amino acid, or what are they doing in the formula?
Yes, they're there. Yeah.
So the amino acids that we're applying to the soil, to the plant, are being broken down by the microbial activity in the soil and then reconstructed intracellularly. Is that essentially what?
Well, that's one way of doing it. Yeah. Plants and microbes interacting with each other can build amino acids, build proteins. Right. Plants might even produce amino acids and send them out through plant exudates in order for they get eaten up really fast, by the way, by microbes. They don't last long in soils. But humic substances stabilize them momentarily so everybody can kind of do what they have to do. So I don't know if that answers your question or not, but what we do is we supply amino acids to the soil and just sort of let the soil do what it needs to do to get that eventually up to a high grade material called protein.
So let's go, just so we can qualify this and define this. I mean, we're hearing a lot, and certainly in our industry, about amino acids. A lot of folks are coming out with amino acid this and amino acid this. And I'd be willing to bet if we were to test, everybody in the industry that's using these things, that probably wouldn't truly understand why. So, in our case, we use amino acids to help stabilize products. But, a lot of folks talk about their foliar uptake. But for the most part, these amino acids are these constructed minerals that hit the soil and then are broken down and can be brought into the plant as the building blocks for the plant's needs. Is that, am I explaining that well, or are we still missing
I think you just explained what some people are doing with amino acids. We're taking a lot different approach with amino acids.
Yeah.
Their approach seems to be simplicity again. Amino acids are pretty expensive. So just how much glycine do you want to put on, or proline, or any of these others that are great at reducing stress, for example? And they're also plant food. They're also microbial food. Our approach is a little different. We look for naturally occurring sources of amino acids.
Right.
Then we bring in like 25 of them instead of 1, 2, 3.
Actually, there's 26 on the full.
Okay, I miscounted.
And we tested. I mean, we took a product. This happens to be, I think, C3. Which is the base of all of our Foliars Plus, but we took that to the lab and came up with 26 complete amino acids in the formulas that you and I have put together.
Mm-hmm.
And the one thing, and let's talk about this. You talked to us about the G's, the glycines, being probably some of the most important, and that's some of the highest concentration of the amino acids that we found within our products. What's the benefit of that G series of amino acids?
The primary benefit, besides everything I already mentioned, we see, I should say, we intend to see more stress resistance. These materials are renowned, amino acids, especially the glycines, are renowned, glutamine. They're very renowned for reducing the effects of reactive oxygen species, ROS. ROS are a response by plants to try to overcome a problem that they're having. It could be something invading them. It could be any number of things. And these ROS are intentionally produced by plants to zap something because they're highly toxic. They're highly inflammatory. One problem is, what do they do when they don't need them anymore? How do they get rid of them? That's where these glycines come in. And proline, there's a number of amino acids that enter into regulating the amount of ROS that's in the system and get it back down to a more balanced level that's more tolerable for the plant. Very, very critical to ROS depletion, actually control of ROS.
Okay, very critical. And I kind of want to make a correction that if they're in the liquid products, they're in the Foliars Plus products, they're not there just to hit the soil and then get munched up by microbes. We're talking about plant response, ROS response in the plant.
Yes.
So they really are a direct application to the plant to be taken in completely, not broken down in the root zone, taken in completely by the plant.
Yes, and we're not applying one, we're applying 26. So
That's right.
That smorgasbord of amino acids is there for the plant microbes to do what they need to do to get the job done, whatever it is they do well. You might get criticized on the concentration of those. And has anybody ever brought that up? They could say, oh, you don't have very much in there.
Well, we don't have very much in there. We don't have very many minerals in there in terms of, parts per billion, but they're there. And
that's because that's the way nature is. Nature provides a smorgasbord of materials, none of them in extremely high concentrations.
Yeah, that's been a common criticism of a lot of this conversation, is that these minerals are needed in such little amounts. And, we're literally looking at parts per billion, and it could be a very small number of
Yep.
parts per billion, but
Mhm.
the bottom line is that it's there, and when the plant needs it, we're supplying it. And let me ask you this: when we spray these minerals through these Foliars Plus products, do they stick around in the soil, or are they just completely broken down or leached out? What happens once they
The humic substances that we put in every product help preserve. They help stabilize these materials. Otherwise, they're very unstable if they were just released and allowed to go out there totally on their own. And the humic materials are also very well known to stimulate biological activity. So we have a nice interchange, a nice synergy going on there.
So we're both holding the minerals and we're providing a haven for biology to really help make it more bioavailable by having the microbes. So, I mean, that really does address the importance of what we've been doing for all these years, which is making very big, diverse, complex formulas. And that's kind of why we do that, is because these things are so important. And I guess it's fair to say, Lawrence, that we may not know what the specific function of some of these metals might be, or these minerals, but we're kind of learning that there is a function. And I think what we're hearing from you is that we're probably going to know more about this, as more research is done across the world. And I'm looking at the encyclopedia of metalloprotein, so I'm assuming that there's a lot of I can't even say that bloody word, but it sounds pretty impressive.
I should mention that this is just one source of that information.
Right.
The literature is full of information, published papers and articles. This is put together into an encyclopedia. The knowledge is so well established.
Well, that's important, and I think that's what we're trying to bring to the table.
One of the points I like to make is that all these 75 naturally occurring elements that I've been talking about are detected in soils. So how much is there? Okay, what's their concentration? I've got a pie graph up on the screen right now of all of the elements that are found in soils. Now, the problem with this graph is it's a mess.
I'd say
silicon takes up 70% of the pie. Okay.
Our silicon sisters friends would love that. 70% of your pie is silicon, but that's the largest mineral found in the crust of the earth.
Oh yeah, it's literally everywhere. Aluminum shows up on this pie chart as probably the next largest one.
10%.
So the best way to do this graphically is get rid of the silicon out of the graph. And that's my next graph. Here's the majors that are in soils. Carbon, major component. Calcium, iron, potassium, magnesium, manganese, sodium, phosphorus, sulfur. Strontium is a major element in soils. Titanium is a major element in soils. Zirconium is a major element in soils. And then aluminum kind of takes the biggest chunk. There's 17 major elements in soils, and I define a major soil element as if it is found in a concentration greater than 100, we'll call it parts per million for your audience. I have milligrams per kilogram on there, but it pretty well translates out to parts per million. So that's my definition. And
so you're telling me that there's 17 essential elements? Is that what we're saying on this chart?
I'm saying that if we analyze a soil and we only quantify, list all the elements that are greater than 100 milligrams per kilogram of soil, then you end up with 17 of those. And the surprise here might be zirconium, titanium, silicon. Well, I don't know what that is back there. I can't read it. And sulfur and sodium's in there. So there's some in there you probably never heard of before.
What is Ba? Barium?
Pardon?
What is Ba?
Oh, barium. Barium makes it because it's at 421 parts per million. Fluorine makes it because it's at 336. And chlorine just makes the grade at 100 parts per million as a major element in soil systems.
So we're suggesting that even a plant material needs some level of chlorine.
Well, according to this graph, if you believe in the if you believe in the proposition that the more you pour on, the more you get out, then plants should be highest in aluminum. Second highest should be iron in plants. So plants should be made out of aluminum and iron according to that logic.
Right.
That's right. If you think that nature doesn't have any control and it's just going to take up whatever there in the concentration that it's there, then according to that logic, you would find more aluminum than anything else in the plant.
So the
That's
plant can actually create, I'm going to use that word again, I'm going to get in trouble, balance intracellularly so that it protects itself from, aluminum in particular.
You can drop the word balance and use the word homeostasis instead.
I like to use the word homeostasis.
Okay. That's what you're talking about is homeostasis. If plants did not demonstrate homeostasis, they would be full of aluminum and followed by iron and carbon. Well, carbon is probably, there's a lot more carbon than any of these in a plant. But anyway, I think we made our point. There's 17 of those. Oh. Then the next slide that I have up here are any element found in soil between 100 parts per million and 1 is called a trace element. There's a lot of them in this pie. The pie chart's very, very cluttered. The majority of the elements that are found under that definition are rubidium. Remember, I told you that was connected to potassium, vanadium, zinc, cerium, chromium. Getting your chromium lately there?
I take chromium every morning. Yes, absolutely.
That's a good idea. Yeah. Okay. We have copper, lanthanum, neodymium, nickel, and lead. Lead is in there. And
now, in this particular chart, and these are small numbers, but you've got a couple of the essential, the 16 essentials, zinc and copper.
Zinc and copper in there.
And they're very small numbers in the grand scheme of things.
Yes, they are. Yeah.
So,
the total count is 37 trace elements. That kind of takes care of
and every one of these trace elements is a building block for enzymes, or as Jerry used to call them, the spark plugs for building enzymes. So they create this ability of the plant to pull enzymes together. Now,
I would say that's the theory, because I've been accumulating information on all these elements to see what role they do play in biological systems, and it's not a full chart yet. I don't even know what I'm up to. I'm up to 33 or 35 or something like that.
Yeah. Now, if we were to tissue test a blade of grass, would we find all of these in there?
If you extract it with boiling nitric acid, then the answer is yes, you will find them. Some of them may be non-detect in grass because grass has its own peculiar elemental profile, its own way of performing homeostasis. So I'm not going to guarantee that you're going to see them all on a blade of grass, but you'll see quite a few. Now, if you go over to asparagus in the garden, you're going to get a different answer.
Right. Let's hope so. But we can make the assumption or jump to the conclusion that if they're there, somehow, some way, the plant is utilizing it. Is that a fair statement? If you
believe in the statement that nothing happens by chance.
I believe.
Then the last slide I'm showing up here are what I call ultra trace elements. They are defined as anything in a soil system that's less than 1 milligram per kilogram. Now, 1 part per million doesn't sound like much, but to a microorganism, it's there and it can be utilized.
Good point.
And much to my surprise, antimony takes the big number here, and thallium. Both of them are very, very toxic materials. And they just so happen to be in extremely low quantities. And the rarest element on the face of the earth in soils is way, way, way down at the bottom, rhodium, 0.0001 part per million. But it's there.
Do we know anything about rhodium?
Yeah, it interacts with cobalt.
So even in that incredibly minute amount, that mineral has a potential to be a building block for a physiological function within a plant.
Yep.
Amazing.
And I got to tell you a story about lithium. This is just for fun. Okay. Now, if you look on the periodic table that I have up on the screen here, lithium is the very first element right below hydrogen. That means it's an extremely low density material, very light material, and it is found in soils at about 20 parts per million. Not bad. It's
Yep.
actually, that meets the definition of a trace element. Okay, so I just show its position on the periodic table because later I'm going to show how that actually is important. The position of a material on the periodic table is very, very critical. Now you're just asking about rhodium, what it does. Notice rhodium is directly below cobalt. There's cobalt, go one square down, and then there's rhodium. Okay, that interrelationship is very critical to the metals across this whole chart. Okay, so I'll get into my point. I have a drawing of this, an outline of the state of Texas here. And there were 2 studies done in the state of Texas. And these studies were about the correlation of lithium in potable drinking water and crime. This is so good.
Yeah,
Okay,
be careful.
these scientists actually said the upper left-hand picture that I'm showing on the screen here would be the upper northwest half of Texas.
So you drew a diagonal line across the state about halfway down.
About halfway down, right? Splitting the state of Texas in half, but not horizontally. There,
Right.
It kind of runs on an angle.
Right. It's
Okay.
about a 7-2.
So northwest of that drawn line there, lithium in drinking water was less than 60 micrograms per liter, non-detect. It would be the other term for it. Below this line, the average lithium in drinking water was substantially more, 120 micrograms per liter. Now, I talk about small quantities. A microgram is a millionth of a gram, or in this case, they detected 0.12 parts per million in the drinking water in Texas as an average in that.
And this was naturally occurring? Is that
Naturally occurring
Naturally
potable
occurring.
drinking water, groundwater, out of wells,
Mhm.
from wells. An inverse correlation, which means that if you have more of this, you have less of that. If you have less of that, you get more of that. An inverse correlation between the drinking water lithium level, in other words, if it went up or if it was down, the correlation was below this line, there was substantial difference in schizophrenia, psychosis, neurosis, and homicide. We're
Wow.
talking about something in drinking water at 0.12. So I hate it when these people say, oh, there's not enough in there to make any difference. Well, how about 0.1 of a part per million having this effect on the human population? Schizophrenia, psychosis, neurosis, homicide. They ran this study again, and they found out that there was, again, an inverse correlation between suicide, robbery, and drug arrests in the state of Texas, depending upon the amount of lithium in the water.
Have they done this kind of a study in other places in the world?
And it was recently confirmed
Ah.
in the EU.
Sorry,
Okay.
I just laid that one out for you, didn't I?
My gosh.
Now, you know darn well, you know me really well. I'm not going to read one study and say, hey, you guys, look at
You know
this.
that.
I'm going to show you the second study. I'm going to show you the third study. Who knows? Maybe there's even a fourth one. But after you get 3 studies that say the same thing, you're on a career path, you might want to go do something else.
Well, the only thing
Okay.
I'm not getting about this is that I thought that lithium was a drug to numb you from psychosis and neurosis. And
Isn't that
You're
what they
giving a
give?
very good example of how higher concentrations have a totally different impact than low concentrations. At
right.
extremely low concentrations, they saw a difference in human behavior.
Right. But then, I mean, I'm not wrong in saying, like, if you are diagnosed with schizophrenia, you often get prescribed lithium. Is that right?
That helps. Yeah, it helps.
So if you get given a lot, it can then maybe.
Where do you think the drug companies got the idea to do that?
Ah, that makes sense now, doesn't
You're
it?
looking at it. You're looking at
Studying
it.
nature again. What a
You
concept.
know what else was discovered in Texas? They found out that the higher the amount of stannous fluoride, I didn't say fluoride, I said stannous fluoride, tin fluoride, the amount of tin fluoride in the drinking water in wells in Texas was also inversely related to tooth decay. And that's where we nowadays they just say, oh, that toothpaste has fluoride in it. That's not quite the way the science panned out. The science said stannous fluoride
And it was naturally occurring again in some of those
is
communities
the one that
where
had
they
the
lost.
effect.
Yeah.
Yeah. Not stannate fluoride, but stannous fluoride.
Yeah.
Okay,
Interesting,
different. Then I ran across this study some years ago, which on the screen just looks like a mess of dots and it's very boring and nobody really wants to pay attention to it. But on the left side, this study done by Tyler in 2004, he was looking at ionic charges and radiuses and things like that. But on the left side of the chart, he plotted the root to soil elemental ratio. And on the bottom of that, he plotted the log 10 ratio of these elements, bunch of elements, 50 of them. He plotted 50 elements. And this result of this one study strongly suggests that there's no correlation between the concentration of elements in soils and how much this root soil ratio ends up being. And this R squared up here in the statistical result is 0.0097, which says something other than the concentration of elements in soils caused these plants to take up these elements. That's what that R squared number says.
The suggestion is what?
Pardon?
What are we suggesting it is that's taking them up?
At this point in my gathering information, I'm just looking at this going, really? Then I found another study by the same guy in this case, where he plotted the soil concentration against the root soil ratio over here on the left. And again, the R squared of this slope, here's the slope of the regression analysis, says there's something going on here besides concentration, soil concentration. That R squared at 0.0076 is, it's not soil concentration that's causing plants to take up these elements. And what got my attention was this one right here, cesium. This chart says cesium is enriched compared to the amount of cesium that's actually in the soil. And then this next dot is rubidium. Rubidium is enriched relative to the amount that's found in soil. And I've been saying rubidium and potassium are always correlated. Ta-da! There's potassium enriched in this. And then you go, what was this? This was a beech tree, this Fagus in a forest pod soil. So I'm going, I wonder if I could just duplicate this experiment. I mean, you could go out and dig up soil and actually measure how much is in the soil and then run a correlation analysis to how much a plant takes up. Oh, just before I jump off of that, mercury, silver, and cadmium, horribly powerful toxins in relatively low concentrations, are depleted in this study. This plant somehow enriched its leaves with cesium, rubidium, and potassium, and actually kept the mercury and the silver and the cadmium down in its uptake. It regulated that uptake somehow. That's intelligence.
Pretty
Okay.
Intelligent.
So why not just duplicate what this guy did? Oh, by the way, before I jump into that, did you ever do the Ringers experiment when you're in college, high school? Did you ever do this one?
No, but
I did this one in high school and college, and we put a battery on these frog legs and they went like that. And the Ringers experiment of 1884, the way it was told to me, it was because potassium channels through our muscles. And if you look up the real experiment, Sidney Ringer, 1884, investigation regarding the action of rubidium and cesium compared to potassium. His experiment was about potassium, rubidium, and cesium, not just calcium. Wow. And look at where they are on the periodic table. Calcium's on top. The very next square down is rubidium. Very next square down is cesium. You can look at any point on the periodic table. Now this applies to metals only. That whole left-hand side. Any point on that periodic table says, if you find potassium in nature, you're going to find rubidium. You find rubidium, you're going to find potassium. You find rubidium and potassium, you're going to find cesium. They're all correlated with each other. And that 1880-84 experiment was about potassium, rubidium, cesium in our muscle system. Now, somehow, as the years went by, we totally overlooked the other two.
Interesting.
Okay.
Well, we're running out of time, so let's see if we can come to a wonderful conclusion here.
Okay, so I asked the question, what is the correlation between nutrient concentration and leaf nutrient concentration? Performed this experiment on 40 different cornfields. The soils were sampled right next to the roots. The leaf stages was at R3 and R5, which simply means There was no fertilizer being applied, so that was not a variable. These plants were totally dependent upon what was in the soil at this stage of their growth. Both the soil and tissue samples were analyzed in the same lab and analyzed 44 soils. Here's the data up on the screen. At any glance at any of this data, it speaks loudly. What it says is that there is no correlation between soil nutrient concentration and the actual amount that a leaf would take up in corn. And here's the data, very technical, but the two bivariants were nitrogen, phosphorus, and potassium, for example. Pearson's R correlation analysis, there isn't any, and the P values say it's reliable data. So I duplicated Taylor's experiment, got the same answer. No correlation whatsoever between how much you pour on and what the plant takes up. There isn't any, non-existent.
So what's the answer?
The answer is balance. And this
biology.
is the dumbest thing I think I've ever seen.
Oh,
do you have any idea what this is called?
No.
This one says how soil nutrients are governed by soil pH.
Yeah.
Well, the first time I saw it, I said, they only went to 4 pH. What about the rhizosphere, which is 2? They totally ignore biological activity in this chart. So obviously it's not right. And there was a recent paper published just recently poo-pooing this pretty darn good. There is no relationship like this. This was printed. This was published as the soil reaction influence on availability of plant nutrients in the plant in the Soil Science Society of America Journal back in 1947. And it was an opinion article. There was not a single reference made in this article. There was no scientific research to back it up. Truog said, "This is my opinion."
So what we're looking at, Lawrence, is the pH chart that's pretty common in every soils book. In fact, I've used it. I think it's even in Jerry's book. But the implication here is that at the pH between 6.5 and 7.0, you have the potential for maximum potential nutrient uptake, because below 6, you lose in macros, above 7, you lose in the micros. That's essentially what that had been used for. But what you're suggesting is even that suggestion?
It's invalid. There's no scientific basis for even using that chart. And there's no references within the paper. It was an opinion paper published in 1947 by Professor Truog.
Okay.
That's all it was, in his opinion.
Right. And then the next slide you have here is the
no research to support this, none whatsoever. And there's absolutely no basis for this. This is von Liebig's
big
law
lie
of the minimum. I mean, it doesn't work that way. It doesn't work that way.
What he's showing here on the slide, for those of you that are listening, is the von Liebig's barrel, showing the minimum, the limiting factor here. And you're suggesting even this is...
You can go out and run the same experiment I did. You can dig up a soil and you can dig up tissue. You can run 40 samples for statistical reliance, and you'll come up with the same answer I got.
Interesting.
Yeah. So you probably don't want to hear about rainwater.
Oh, go ahead. We're
okay.
Way overdue here.
Okay. I had a client that said, I said, I think, he said, look how complex our world is. Look at how complex our soils are. Look at how complex everything is. He said, look at how complex our atmosphere. He says, hey, what's in rainwater? I said, gee, I don't know. I couldn't find any data on it. So pulled 61 samples that year. I think it was 2014. It was one rainy year to pull 61 samples. Okay. The crazy thing was the data was very, very variable. It was extremely variable. There's my little apparatus that I used. It's a Chemex. It's a Chemex jar, and with bird netting around so they wouldn't poop in it. I detected 71 elements out of that database. Only 5 elements were non-detect out of the 75. There was one other element there. I think it was xenon or something that was non-detect. Rhodium, the rarest element on the face of the earth, was a real shocker. It showed up.
There?
It was there. Yeah,
wow.
My gosh.
In rainwater.
So I boiled it all down into a statistics chart. The chart says that the majors are all in green that I detected, including rhodium. Rhodium was in my 3rd quatrile of statistical data. Boron, aluminum, silicon, phosphorus, sulfur, rhodium, indium, extremely rare as rhodium, bismuth, that was a surprise, 2 rare earths, And then the usual suspects, sodium, magnesium, potassium, calcium, and yttrium, which is also considered lanthanide, were all there in substantial quantities. Everybody else fell in behind that. So detected 71. So how many elements does it take to grow a plant?
I love it.
Wow. And have we concluded that?
I love it.
I don't think there is an answer to that. I don't believe
No.
there is an answer.
Wow. I mean, that's fascinating, to be honest with you. And again, I think I think there's a lot of conversation still yet to be had on this subject, but clearly, the old adage of the 16 essential elements, it just doesn't make any sense. I mean, the plant's a lot smarter than that. And I think that's why we've been so active in making sure we're bringing in raw materials to provide all these essential elements that we'll call essential elements. But again, if you look at this particular list, and we have this published on our website, but in the products that you formulated with us, we've got 75 minerals and 26 amino acids, all of which are going to ultimately feed the plant. Lawrence, I hate to say this, but we're way over time, but we could do this for another hour easily. And which is why we have you back on the podcast so often, because there's so much to talk about. But I mean, Jack, have we concluded anything? I mean, I think all we were really trying to accomplish was, at least throw out the thought that maybe the plant is looking at a lot more than just some of the basic stuffs. And certainly that moron theory has been debunked of, all you need is moron. I mean, there's a lot of talk in our industry about, minimum levels. And I think there's a lot of validity to that. But, it really goes back, Lawrence, to our concept of biological soil management and remineralization, which is what, we started this whole conversation back in 1988 on, is providing enough of this mineral and then producing a biological environment where that mineral can then be, brought into the plant. And again, basically the definition of biological soil management. I mean, if we were to boil this down to a couple terms, is that a fair assessment on what we just went through here?
Yes. Yeah. That's why people like buffets.
That's exactly
Oh, I
why.
like that analogy.
Yes. Yeah. Everybody eats a little bit differently and has different tastes, and so do microbes, and so do plants.
Everybody.
I'll give a whack at a little synopsis and conclusion. I mean, the big crux of the whole podcast was that nature doesn't leave anything to chance.
Yeah.
That it's not uncontrolled. That nature's controlling its uptake. We kind of also broke down the 75 naturally occurring elements that are, on the buffet line, so to speak. We spoke about the lanthanides, the rare earth minerals, and how they were to be interchanged with calcium. And I kind of described them as a super calcium, or rather that when the plant is in extra stress, it really calls upon these rare earth minerals, or lanthanides, you refer to them as quite a bit. And that stress is all kind of what a good structure strengthening would prevent. You talked about a frost example, which was really interesting because calcium is much less bioactive at low temperatures, but some of those rare earth minerals were more active at the lower temperatures. We also got a good conversation about how amino acids again are used in that stress situation, and that in that they are natural structures, they do have within them some of those cofactors or those trace elements and ultra trace elements that are already built within them. So the fact that they're in our formulation, they're providing that, and they're providing it in nature's language already built in the amino acid. So I guess, the big one being that nothing is uncontrolled.
Yeah, Mother Nature has a plan. I mean, I think this really, it's got to get everybody thinking that the concept of all you need is urea is just invalid. It just doesn't make sense. I mean, not only is it going to build thatch and destroy soil structure and take away carbon from the soil, but it's not providing these essential elements, these minerals, these trace elements that the plant clearly needs. So, I mean, I think we've certainly beaten this horse pretty well, but I think we certainly made the case that there's a lot more to Mother Nature than just, putting down some urea. And I think we'll leave it at that, Lawrence. And I'm going to say thank you very much for your time. I apologize for us going a little long, but I'm always thrilled to have Lawrence on the podcast because he brings to us a freshness and a science that is, hard. And I realize that a lot of this conversation is probably a little difficult to digest, but take it in pieces. You can always come back and grab a clip here and there, but this is what we go through every week when we sit down with Lawrence as on our team of agronomists, and we record it because there's a lot to take in. But as this starts to sink in, and you start to really make the concept, understand the concept that these minerals, that there's so much more complexity to what we're doing in terms of fertility. And that's why we've been so focused on what we call biocharge, this whole package of amino acids and minerals. And it's so important to us to remineralize. And again, that's an adage that Jerry Bernetti taught me very early on in my career. And it still rings true to this day. And Lawrence, again, I got to say thank you. And Jack, I want to say thank you for joining us. We will probably have you back on sooner than later, Lawrence, and we'll see you every Monday. And that's why we record these things. But again, I can't thank you enough for your time and your support for all you do for us. And I don't know what we would do without you, quite honestly. So I'm going to say thank you for joining us. If you're not a subscriber to the Earthworks podcast, please be, so you can be notified when Lawrence gets back on the podcast with us, which probably won't be more than a few months, as we tend to have you back and forth all the time, Lawrence. And Jack, thank you. And Lawrence, thank you so much.